Thanks everyone for joining today's webinar on advancing our understanding of the hallmarks of cancer using live cell analysis. So during this talk, I'll touch on the hallmarks of cancer, introduce the IncuCyte system, and then talk about how we can use live cell analysis to investigate the hallmarks of cancer. In our search for ways to treat cancer, it's vital that we have the ability to distinguish cancerous cells from normal ones. So to this end, Hanahan and Weinberg first described hallmarks of cancer in 2007. These hallmarks describe the characteristics of cells that have transformed from normal to malignant. But cancer as a disease isn't caused by one cell suddenly becoming bad. There's a number of dynamic and interdependent processes that happen through time that enable tumors to form. As well as the plasticity of cancer cells, their total environment must be considered, and this means that both cell-to-cell and cell-to-extracellular matrix interactions are both important. As our understanding evolves, more hallmarks are being added, and now at least 10 are being described. For us researchers to fully understand these processes, we have to use the most appropriate and relevant methods that are accessible to us. So ideally, the systems that we use should be versatile enough to be used across different biological models. They should be non-perturbing so that we can use even highly sensitive cell types, and they should give us robust and reproducible data sets. Live cell analysis refers to the continuous measurement of cells through time. The IncuCyte system sits within a standard cell culture incubator and acquires phase, fluorescence, and bright field images from underneath your tissue culture flasks or microplates. After every acquisition, the images are processed and cells or 3D objects are segmented, and this provides us with real-time data. The whole process is completely non-perturbing to your cells and to experimental setup, so we can monitor your whole workflow from start to finish. The health of our cells can be monitored as we look at cell morphology and measure proliferation, and this makes sure that the cells that we're using are as healthy as they can be and behaving as they should. When the cells become confluent, they can be passaged, and different treatments can be applied to get them ready for assay. This could be differentiation, synchronization, or the formation of 3D spheroids. Finally, we can perform kinetic assays where we examine cell behaviors, including functional assays or response to cytotoxic compounds. Image acquisition is non-perturbing and the cells are kept within a physiologically relevant environment, so our experiments can last for as long as we need. This can be hours or up to days or weeks. Data analysis happens in real time, so we can make data-driven decisions about when and how to proceed with treatments. And with kinetic assays, there's no need to define an endpoint, because the cells are being continuously monitored, so no critical events will be missed. The first hallmark of cancer we'll investigate is selective proliferative advantage. Cancer cells display a high rate of proliferation, and this gives them a competitive edge over normal cells. They experience sustained proliferative signaling and divide more often. It's therefore critical that we can get quantitative data on cell growth. Using live cell imaging, we can get fully kinetic proliferation data from cells growing under different conditions. The time-lapse videos here show the growth of 4 different biological models. On the left, a 2D cell monoculture is captured using phase images. As the cells grow and divide, they become more confluent and can crowd together. Bright field images can be used to capture single spheroids in a scaffold-free environment, multispheroids grown on a layer of extracellular matrix, and organoids which have been embedded within a dome of extracellular matrix. These larger objects tend to increase in size over time as the cells grow, and certain types of organoids can also become darker as they mature. With pancreatic organoids shown here, a dark center is a sign that the organoids need to be passaged. By identifying the objects of interest, and that can be single cells, spheroids, or organoids, we can turn these images into quantitative data. This is achieved by segmenting the objects, and the yellow outlines in the images above show the boundaries of the identified cells, spheroids, and organoids. With the IncuCyte, segmentation can be set up within the software. Then after each image is acquired, the segmentations will automatically be applied, and the image will give us an updated data set in real time. Within 2D cell cultures, we measure cell count and the total confluence over time, while spheroids and organoids can be quantified using bright field data. The way that cells respond to stress is critical to their survival. Either damage can be repaired or cells will undergo programmed cell death. As the cells proceed through the cell cycle, they have to pass through a number of checkpoints, and these are points of quality control that mean the cell is healthy enough to proceed to the next stage. In response to stress, these checkpoints can be activated and the cell will either become senescent as it tries to repair the damage, or it will induce apoptosis. Stress comes in many forms and can include a lack of growth factors, oxidative stress, DNA damage, or defects in the mitotic machinery. As we've noted in the previous hallmark, cancer cells have a higher rate of proliferation than normal cells. This means that they proceed more rapidly through the cell cycle. They divide more often and have a higher mitotic index, which is the proportion of cells in any population that are mitotic. Therefore, the cell cycle and mitotic cells are an attractive target for drug discovery in oncology. Here we demonstrate the use of advanced label-free classification, which can identify mitotic cells in a phase image by their morphology. The images here show that in the presence of nicotazole, there's a higher number of mitotic cells as compared to the vehicle control. These are identified in the bottom row of images by the segmentation masks, where pink indicates that the cell has been classified as mitotic. The time course of mitotic index reflects this observation. Post-treatment, the nicotazole-treated cells experience a rapid increase in the number of mitotic cells over a 24-hour period, which then begins to drop. Using live cell imaging to follow this kinetic process, we can see that the cells are in mitosis for a longer time than usual. Nicotazole stabilizes the microtubules that are necessary for spindle formation during mitosis. This is a form of chemical stress that can activate the spindle assembly checkpoint, and by extending the time in mitosis, p53 can be activated, which then increases pro-apoptotic signaling. By segmenting cells, we not only have access to data on cell count and morphology, but we can also measure the fluorescence within cells. Here we show that by using cells that express our IncuCyte cell cycle reagent, we can look more closely at cell cycle phases. As the schematic shows, cells expressing this reagent display a green fluorophore in SG2M, red or orange in G1, with the two transition phases being yellow or non-fluorescent. The top image shows that in healthy growing cells, there's a mix of all four populations. Treatment with compounds that target the cell cycle can induce cell cycle arrest, so the proportion of cells in each population will change. Cisplatin is an anti-cancer compound that binds to DNA. It forms adducts and induces DNA damage stress. Low concentrations induce arrest at the DNA damage checkpoint in S phase, and this results in a high percentage of green cells, as we can see in the middle image. The time courses at the top show cell cycle phase. Cisplatin treatment induces a time and concentration dependent increase in cells arrested in SG2M between 12 and 24 hours. At the same time, the percentage in G1 decreases. The bottom time courses show that at higher concentrations of cisplatin, proliferation slows, which causes cell count to plateau. In this example, we have included our Annexin 5 cell health reagent, which displays a blue pseudocolor in dead cells. At the highest concentration, apoptosis is induced, and the percentage of apoptotic cells starts to increase, but not until after the 24-hour time point. The bottom image shows the dead cells binding to the annexin reagent, indicating apoptosis at 48 hours. In order to survive and proliferate, tumors can induce angiogenesis, which is the formation of new blood vessels. These blood vessels supply oxygen and nutrients to the center of solid tumors, which allows them to grow in size. Our angiogenesis assay uses endothelial cells expressing GFP in co-culture with unlabeled fibroblasts to create a 2D in vitro model of tube formation. The video shows GFP HUVEC cells forming a tube network in response to VEGF over a 7-day time frame. Using a bespoke analysis, we can distinguish single cells from tubes and quantify the formation of the tube network. The image on the top left shows the green fluorescence channel of a fully formed tube network at 7 days. Underneath is the segmentation mask derived from the analysis job. As the time course shows, tube length increases in a time and concentration dependent manner in response to VEGF stimulation, and we can derive EC50 values for these growth factors. With this model, we can also measure inhibition of angiogenesis in a high throughput manner. Anti-angiogenic therapies include monoclonal antibodies against VEGF and other growth factors, and here we demonstrate the action of the anti-VEGF antibody Avastin. Increasing Avastin concentration stops the tubes from forming, so we see a reduction in the total tube length. Metastasis is the leading cause of cancer-related deaths. It's not just a single process, but a series of connected events where highly plastic cells can transform, and this often makes them more motile. They move out of the tumour microenvironment, invade through the extracellular matrix, often into the circulatory system, where they can move more freely through the body. During the next stage of metastasis, the cells invade again, back through the vascular basement membrane, where they can start to grow and form a secondary tumour. Being a dynamic process, live cell imaging is a really useful tool for measuring cell motility. With time-lapse imaging, we can monitor the progress of cells as they move. In the video shown here, we can see cells moving into the wound area that's been generated by 96-well Woundmaker. These images are translated into quantifiable data and can be used to determine the effects of anti-invasive or anti-metastatic drugs. Here we have two in vitro models showing cell motility. On the left, cells are invading into extracellular matrix, and on the right, they are migrating into a wound area. Although these both show HT1080 cells, we can observe the difference in morphology between the two models. Migrating cells are fairly flat, whereas invading cells have a more branched phenotype. Looking at the time courses of wound density, we can also observe that migration occurs much more quickly than invasion, with the wound closing in 12 hours by migrating cells, whereas invading cells don't close the wound until 60 hours. We also demonstrate different pharmacology between these two models. In the presence of blebistatin, invasion is reduced through time, but migration is not affected by the same concentrations. We can also examine invasion using a three-dimensional spheroid model. The video shows cells branching out from the central spheroid body, and over a 9-day period they invade into the surrounding extracellular matrix. As well as measuring the central spheroid body, we can also measure the area of invading cells. The time course of invading cell area on the right compares different cell lines and shows us that the U87MG cell line is much more invasive than both A172 and HT1080 cells. Inhibiting this invasive behaviour of tumours is the way that we can try to stop or slow cancers from spreading throughout the body. Live cell imaging can be used to evaluate potential therapeutics and gain information on their mechanism of action. Our vehicle cells show strong invasion into the ECM, with a wide area in the blue mask representing the area of invading cells. Cytochalasin D inhibits actin polymerization, which stops invadopodia from forming and reducing invasion. Although it's a good anti-invasive compound, we can observe that the central spheroid body is still quite large. PP242 is an mTORC inhibitor and is known to be cytostatic. But while it inhibits the growth of the central spheroid and reduces the amount of invasion, treatment with this compound yields the same ratio of invading cells to whole cell area as the vehicle. The metabolic function of cells can change as they undergo neoplastic transformation. Cancer cells can have a higher rate of metabolism to sustain their rapid growth and plastic phenotype. Therefore, the ability to identify dynamic changes in metabolism is critical. The IncuCyte ATP assay enables real-time evaluation of ATP levels within living cells. This assay uses a FRET-based biosensor, and when ATP binds, the FRET pair moves together and the fluorescence increases. The images here show SCOB3 cells expressing the genetically encoded cyto-ATP biosensor and the non-binding control sensor. The cyto-ATP sensor responds to the presence of ATP, and as the image on the left shows, the fluorescence ratio is high. In the control, mutations stop ATP from binding, and this provides us with a baseline level of fluorescence. Using these two sensors together, fluorescence ratio calculations are all automatically performed within the IncuCyte software after each image acquisition, allowing users to very easily view the results. The image acquisition and analysis software have been purpose-built and designed to be user-friendly and accessible. Here we can see the effect of glutaminase inhibition on 3 different breast cancer cell lines. Triple-negative MDA-MB231 cells experience a sustained drop in ATP in the presence of the compound CB839. MCF7 and ZR751 cells are receptor positive And while MCF7 show a transient drop in ATP, they recover by 48 hours, whereas the ZR751 are completely unaffected by the same concentrations. Comparing the concentration response at different time points demonstrates why kinetic monitoring is important. While at 15 hours, both the MDA MB231 and MCF7s are affected, by 72 hours the MCF7 have recovered, and the CRCs show that only the MDAs have a sustained drop in ATP levels. The immune system plays a complex role at all stages of cancer initiation and progression. Immune cells are part of the tumor microenvironment, and they can induce inflammation, which in turn promotes other hallmarks such as metastasis and angiogenesis. Immune cells can also work in our favor, and by sending the right signals, we can use them to attack cancer cells. Using live cell imaging, we can visualize and quantify the process of tumor cell killing by various immune cells and probe the mechanisms that allow this process to occur. CD47 is a so-called "don't eat me" signal that's highly expressed on tumor cells. It's thought that this is one of many mechanisms that allow cancer cells to hide from immune cells. Using monoclonal antibodies, this signal can be disrupted, and it allows macrophages to target and engulf tumor cells. The video shows Ramos B lymphoma cells, which have been labeled with the pH-sensitive fluorophore. When macrophages engulf the B cells, the pH inside the phagosome shifts and the fluorescence intensity increases. So we observe an increase in red fluorescence within the macrophages, whereas the free non-engulfed cells show very little fluorescence. The plate view shows the increase in red fluorescence area. In the presence of the anti-CD47 antibody, we observe a time and concentration-dependent increase in fluorescence, but in the presence of the IgG control, no fluorescence is observed. Herceptin is another monoclonal antibody that's been clinically approved for the treatment of HER2-positive breast cancer. Herceptin binds HER2, allowing cytotoxic T cells to target and kill the tumor cells. In this example, we show HER2-positive BT474 tumor spheroids which express a GFP, and these have been treated with a Herceptin. The top image shows treated spheroids on their own, and you can see that they are large in size and highly fluorescent. The bottom image shows the Herceptin-treated spheroids now in the presence of fresh PBMCs. The T cells within the PBMC population kill the tumor cells, so the spheroids are smaller, with lower fluorescence intensity as the cells have died and they stop producing GFP. The time course measuring the GFP signal of the spheroids demonstrates that this killing process is dependent on the Herceptin concentration, with high concentrations of the antibody inducing T cells to kill the spheroids and reducing fluorescence. On the right, we can also demonstrate that this effect is dependent on the target cells. BT474 expresses HER2, so in the presence of Herceptin, the T cells are able to kill, whereas MCF7s don't express HER2, so the presence of Herceptin has no effect. The spheroids continue to grow in size and increase their fluorescence, and the T cells are unable to kill them. As I've mentioned previously, another important consideration is the microenvironment of the tumor cells. And for this, we have to be able to construct biological models that take into account both cell-to-cell and cell-to-ECM interactions. The presence of supporting cell types within the tumor microenvironment can enhance the growth and drug resistance of tumor cells. Cells will also interact with the extracellular matrix, which provides a structural support, and this adhesion can also activate receptors on the surface of cells. In this cytotoxicity assay, we have used SKBR3 breast cancer cells expressing red fluorescence within the nucleus. This gives us an accurate count of just the SKBR3 cells when they're both in monoculture or in co-culture with unlabeled fibroblasts. The cells have been treated with a concentration range of lapatinib, which is a drug used to treat certain types of cancer. In the concentration response curve here, we show that SKBR3 cells in monoculture show sensitivity to lapatinib. But in the presence of fibroblasts, the efficacy of the drug is strongly reduced. The stromal cells are having a protective effect on the cancer cells, so this is a clear indication of why in microenvironment of cells, in this case the presence of supporting cells, needs to be considered within our experimental setups. With these in vitro systems, we can further increase the complexity of our models. As well as comparing cell monocultures to co-cultures, we can include interactions between cells and ECM. In these images, we show MDA MB231 multispheroids on a bed of ECM in monoculture in the top row and in co-culture with fibroblasts on the bottom row. The two models show very different morphology, and the presence of fibroblasts seems to increase the invasiveness. The monoculture multispheroids have in general quite a compact morphology with some invadopodia forming at later time points. In co-culture with the fibroblasts, the multispheroids on the bottom row have completely different morphology. At 3 days post-seeding, they already show an invasive phenotype with multiple outgrowths early on that expand through time. With bright field imaging, we can quantify both of these models. The data here shows that in both monoculture and co-culture, the cells or spheroids grow over time, with the co-culture covering a slightly larger area. So just to wrap up, the IncuCyte platform enables a wide variety of assays to be performed, and it adapts to investigate in vitro models of the hallmarks of cancer. The versatile image acquisition modes enable non-perturbing analysis of cells in monolayer culture, as well as more complex models such as tumor spheroids and organoids. A flexible choice of multiple fluorescence channels, including FRET detection, enables live cell analysis of a wide range of dynamic cellular processes. The automated and bespoke image analysis algorithms simplify data analysis for all of our users. So to learn more about the IncuCyte or any of our validated assays or reagents, please visit our web pages. These have posters and protocols, and you can also download our latest handbook. Thank you very much for your time.